Well, good morning, everyone. A bit late. My apologies. This morning, as I was releasing the exam and looking at some emails, it struck me that we have the fall 2003 recorded lectures. So you have received an email for the dates under that streaming video icon, fall 2003, that correspond to the lectures 7, 8, and 9. So today we're going to do lecture 10. I hope others were as somehow enlightened somewhere along the way that folder still existed. Somehow it slipped my mind that it was there. I would have referred to you to it sooner. Exam 1 is posted. I've increased it to 3 attempts. So there are 3 attempts, 1 hour in between. The exam became available basically at 9 o'clock this morning and is posted till 11:55 on the 19th. I think that's Sunday? Saturday. Let's just go and I'll double check that. Want. So you will see exam 1 posted from today through February 19th, 11:55 p.m. 3 attempts. You get 60 minutes for each attempt. So it's set to go. That should give everyone plenty of time to do it. Take advantage of having the 3 opportunities to go once, give it a check. Again, remember that the academic dishonesty regulations are stated in the syllabus, but you have the authority through various recorded lectures to have blank pieces of paper and a pen or pencil to make notes on the questions, to help you correspond with me, to ask questions about the questions for clarification, and of course to assist you in your preparations to score the highest grade possible. The highest grade counts toward your final grade in the class. So let's come back to the computer and get to lecture 10. One other comment, the day of the posting of the exam is intended as we have today, just to get a recording of the hopefully only one outstanding lecture, and attendance is never required, but I'm pleased to see all of you here today and waiting so patiently for me. I realize that if you hadn't had the chance to view the 2 previous lectures, 8 and 9, that this is a bit out of sequence, but we've been talking all along about the essential need for the development of the circulatory system, and that is presented really in a brief overview in the lectures preceding this one. And we, of course, look at it in much more detail when we look at study organogenesis. We also stated the importance of the developing nervous system, so we have contact between neurons and their target peripheral cells very early on, so they all grow to their final destination collectively and together. And as well, the developing nervous system and circulatory system is going to contribute, and along with everything else, is going to contribute to the rapid growth in the size of the embryonic disc as we had left it. So there is a tremendous amount of folding and reshaping basically the morphogenesis into a structure that is going to have body cavities and more three-dimensional complexities than the simple three-cell disc, three-layered disc that we had talked about. So the 3 weeks, 3 layers, it was what we were working on. We can now take a bit of time to summarize embryonic weeks 4 through 8. Why do we stop at 8? At 8 weeks, we refer to the conceptus as a fetus, at least the embryonic portion of it, correct? So in the 4th week, in the beginning, there's a 3 millimeter straight embryo with 4 to 12 somites. So we saw that first somite appear, and now we are beginning to think of the staging of the embryo, even with reference to the number of somites, the 1 somite stage, the 12 somite stage. So there are many different ways of staging the progression of the development of the embryo. And we really just include them. We have somewhat of an overview, but I don't expect any of us to become experts in that. The neural tube is still open. We know we need to close the neural tube. You all may have recalled that in Texas, not so long ago, there was an epidemic of spina bifida, the failure of closure of the caudal neural pore, and therefore the skeletal system and muscular system and the skin even sometimes doesn't develop over the caudalmost spinal cord area to varying degrees. So the complete closure of the neural tube is really important. And I'm sorry, I need to back up. The epidemic actually was anencephaly. The frequency of spina bifida is still relatively high, I think, in the state of Texas, but it was the spina bifida, right? Anybody recall what was the solution to that epidemic? Shame, 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 shame. Folic acid. Yeah, folic acid. Diet. It wasn't toxins. It wasn't anything to do with the environment. Industries got off this time. It was folic acid. Diet. Strictly folic acid. It's loaded in the Time magazine. All the magazines have that out there. So keep that in mind, that nutrition is extremely important for the maternal nutrition is extremely important for all these developments that we are discussing. And there will be the initial development of the face and neck region. So the area of the developing embryo that's going to become the head, face, and neck starts off flat. It's going to get these finger-like projections that come out and around. They're going to meet and fuse together. There'll be folds in places and everything to form the entire facial region and the neck region. This again represents kind of a segmental development where within each one of these finger-like projections or pharyngeal arches, branchial arches will be another term you'll hear me use. They'll have their own neurovascular structure, their own mesenchyme, mesodermal, more completely differentiated mesodermal cells can be referred to as mesenchymal cells as well. And each of those cell types within each arch is going to contribute to certain structures ultimately as we proceed. So this figure again is taking us through various stages, day 22. So what week is that? Week 4. Day 1 of week 4, right? Week 3 should end at day 21. So day 1 of week 4 should be 22. I'm looking at you for reassurance here. Come on. So the actual size, about 2.5 millimeters. So this is what was stated. We see that the rostral and the caudal neural pores aren't even close to closing. We still have a highly developed neural groove rostrally. Various somites are present as well. And 23 days, we see that the closure of the neural pores or the neural folds to form the neural tubes, the neural tube it has extended quite a distance, I mean, in the illustration, but it's not even quite a millimeter in each direction. So the rostral pore looks larger and is, of course, going to close quite rapidly and will close before the caudal neuropore. And so if there's failure of closure of the rostral neuropore, then there will be anencephaly, and it can have a variety of variations, but basically anencephaly means what? Prefix an means what? No or none, and encephal it refers to brain, so there would be no brain essentially that would develop. But usually it includes craniorachis, which is the incomplete closure of the cranial skull, so the whole cranial cavity with the basically the whatever forebrain there is left would look just like this, just a very flat structure, no cortex formed and things like that. Then on day 24, we already see that there's some folding in the head region and the tail region. So this structure is now wrapping around laterally and in the front and the back and beginning to close off the body cavities around the body stalk. And we see the somites are still present, some limb buds, and the bulging that will appear in the region that we now can recognize as the chest and the abdomen will be contributed by the developing heart and the liver ultimately. So day 25, 26, and then finally to day 28, there's tremendous changes that occur. And as I stated, we'll revisit these again and again. We'll do the organogenesis, and as well, and we study the development of the limbs and the various other systems. We'll revisit all of this information. So the somites are very critical to the formation of the axial structures. Remember that it was the paraxial mesoderm that's going to thicken into these somites, and obviously there's a lot that has to be developed to surround the neural tube and form everything that is there, the entire musculoskeletal and circulatory systems, and of course the nervous component of that for the innervation of all these areas. And lymphatics, don't want to forget that. So here's this marvelous table, the criteria for estimating developmental stages in the human embryo. We're not going to try to go through this entire thing here in class. Obviously, this is something you should review and try to familiarize yourself with as we proceed through organogenesis. We'll revisit some of this, try to use the terminology. No intention of becoming an expert in this, not even in my lifetime, if I were to teach this course a zillion times. Of course, it's all recorded, so 15 years from now, I'll play this back and say the same thing, right? So the age in days with the figure references are shown here. Carnegie stages by number of somites is useful for a while. The length in millimeters is key for identifying this, and of course it's mostly sonography, ultrasonography, that is, we know, is used to make these estimates. What is the, if you would push the button and introduce yourself and your expertise. What's the smallest size that can be detected with the ultrasonography? Push the button and hold it. Well, what's the smallest that you ever remember recording? Push the button and hold it. Okay. I put her on the spot, right?
Right, I know, you did.
Hide your sucker, you can't have food in it.
Now you tell me. We see the fetal pole at about 3 weeks. We see a heartbeat about 5. Yeah.
So the 20 to 21, you're not really seeing the embryo to be able to measure down to a resolution of 3 millimeters. So somewhere, so week 5 is 35 down here. So down to about 10 millimeters or so. And then the main external characteristics are listed here. Sorry to put you on the spot like that. You did well. Excuse me. Then we can take a look at this illustration of the embryo. This is looking at Carnegie stage 10, about 22 days, going through to about Carnegie stage 10 at 27 days in these two panels. What view are we looking at here? In the upper panel, dorsal view. What are your cues to tell you that is true? Somites, without doubt, but definitely the neural fold, the neural groove, and still the primitive streak even being present. And the cut surface, of course, of the amniotic sac. And as well, in the bottom panel, we're looking at the dorsal view as well. And at this stage, we really are just putting these images in our mind. We'll see them again, looking at each organ system. And we start all these different planes of cross-section and illustrations to follow the migration of cells and the development of the different organs, tissues, and differentiating cells within all of those. We don't, with each of these illustrations, we're not trying to extract every minutia of detail and become an expert on everything that we see here. We just want to be able to recognize that at this point, if we have the neural pores still open and we don't have a tremendous amount of folding, there's no limb buds, we're still somewhere within the 3rd or 4th week. Thus, it brings us to this type of an illustration, which is quite obvious for utilization on an exam to label certain structures. We orient first to the neural folds and the neural groove running down the center, and therefore it's a dorsal view. We orient to the cut edge of the amniotic sac, also revealing that it is a dorsal view. Along the midline, we know that the neural folds will be meeting to close off the neural groove into the neural tube, although we wouldn't really necessarily call it the neural tube here, but it is. On the surface of a tube, I guess you can call it a tube as much as if you see it at the cross-section at the end. We see the somites. Remember, the first somite is somewhere in the center, and then the somites begin to develop rostrally and caudally away from that. So there's that axial orientation at the proximate center point of the embryo, and development rostrally and caudally. Rostral develops more rapidly than caudally. Rostral neuropore closes before the caudal neuropore. And the neuroenteric canal at the primitive pit and the primitive groove, although very tiny, is still evident at the very caudal point on this dorsal view of the embryo. So it's obviously very easy just to have this in an image processing software system and delete all of these, have the little lines there, put numbers, and then ask you to identify the structures. And this, of course, is labeling the primitive segments. We would call them somites, but it reminds us of this initial segmental development of the embryo that is lost in humans. But produces, of course, the essential segmental neurovascular supply and the musculoskeletal structures in all of the areas. We don't have ribs all the way down to our pelvis, we know that, but the capability of having ribs develop higher and lower does exist within the system, but there are molecular mechanisms within development, expressions of various genes at different times throughout these different segments that will trigger different developmental features. That, of course, is not a focus of our course, but it certainly would be something that you would have the opportunity to study in great detail if you took the developmental biology course. The end of week 4 then, so this would be basically day 27, day 20, is that right? Day 28 should be the first day of week 5. So the rostral neuropore closes, forming the forebrain. So when we take a flat structure and we curl the edges to make the top or the two edges fuse together, we now have a potential anatomical space. What's contained within that space? Every space so far has been the same answer, extracellular fluid, right? So there's no air within this neural tube. But really, what do we call the liquid within this space within the nervous system? Would CSF help you at all? Cerebrospinal fluid, right? So cerebrospinal fluid is extracellular fluid extracted basically from the plasma in the mature brain once the choroid plexus has developed, but there's plenty of extracellular fluid to be available here. And so basically the amniotic cavity has its fluid within it, right? And it's extracellular fluid. Regardless of the origin, it's still extracellular fluid. So I guess we need to remind ourselves that every time we look at this cut edge of the amniotic sac, it's actually a dorsal view. If we were within that cavity, the amniotic sac, we'd be swimming within the fluid. That fluid is referred to what at the time just before delivery? What do people usually say when they know they're about to deliver? Oh, my water just broke, right? So that water is the amniotic fluid, and of course it will be the fluid that will be initially washing through this fold and neural tube until it's ultimately closed, and then it'll be extracellular fluid generated from the cells lining the neural tube that differentiate into the, what's the type of cell that's found on the choroid plexus? Good question. I'll leave you to think about it. Maybe it'll pop into my head in a few minutes too. Ependymal cell, right? Yeah. It's a type of non-neural brain tissue cell, the ependymal cell, ependymal cell. So the limb buds are evident, the early eyes and ears are appearing. So as we begin to get the segmental development as well of the highly specialized face and neck regions, there will be folds that are indicating these special sensory structures. So in a truly segmented organism, do they have a face and they have a head or cephalad end where food goes in and then the other end where things will come out, but do they have a specialized face? What is the dedicated function of the rostral end of an organism? You got a sucker in your mouth, there should be a dead giveaway there, right? So you've got to find food. What are the two basic functions of every organism? Well, survive first and then reproduce next, right? So survival and reproduction. So the survival part is really what we're considering for the most part in development of the rostral end of organisms. Obviously, there's sensors to find mates and to coordinate what needs to be done to reproduce, but most of the reproduction, as we know, happens at the other end that we've already studied. So all of these highly specialized structures don't just appear from somites. In organisms that are going to develop a specialized head and face for this rostral function of survival, seeking food, and territory, and protection from predators, and all that stuff, we're going to have specialized mesenchymal cells that are going to create the bulges and emerge as these pharyngeal or branchial arches. So the early cardiovascular system, obviously, the heart and the vessels are going to develop very early on. The caudal neuropore closes last, after the rostral neuropore. The embryo is curved laterally, rostrally, and caudally. And all these, as we said, are going to be reinforced throughout the rest of the semester. So we'll build on this information. 33 somites at the end of 4 weeks. And then the beginning of an obvious tail that we know is a remnant of a tail in human primates and other subhuman primates that do not have tails, prehensile tails. It can persist in some individuals with a prehensile tail that's usually removed just at birth or shortly thereafter in the neonatal stage. But what is the remnant of this structure? Basically, what does this little tail end up becoming? Coccyx, essentially, right? So here is this lateral view of the Carnegie stage 13, about 28 days. So it's about 4.5 millimeters long. And even at this stage, is it the linear length along the curve? Is that how it's measured when you have to do it? Or is it crown? Yeah, so it's still the crown rump length here to here? Okay, thank you. So basically the long axis of this three-dimensionally complex structure. So we no longer have a disc, 2 or 3 cell layer at the end of 4 weeks. We have what is recognizable as a complete organism. Nearly all the systems are present for this function, for this organism, this conceptus, to continue its development. We see that in what we recognize as the future face, with the site of the lens placode for the eye and the nasal placode here, we know we have basically the upper part of the face, but we don't have the masticatory or communicative portion of the face yet. So something comes in from down here to fill in that space and complete off the face and, of course, the oral cavity and the lower mandible. So this is the task of the mesenchymal cells that are differentiating and becoming highly specialized in this area of the most rostral somites, and it actually pushes out like this. But what really causes the bulge in there? Other than a localized increase in the numbers of these mesenchymal cells, you're going to have a dedicated neurovascular bundle in there. So there's an artery and a vein and the ingrowing nervous system present within here already, and lymphatics, obviously, somewhere along the way to be laying down. We see the heart bulge on the surface. If you see an image like this on the exam, it's very likely going to be one of these half-tone, black and white photographs with structures labeled, or a line drawing, most likely. I don't think I used very many of these color illustrations. So we are trying to recognize the organ systems beginning to emerge, the limb buds, the upper limb bud shown up here, and the ventricle and the atria of the heart are shown up there. So we can recognize head, face, neck, thorax, upper extremity, lower extremity, abdomen is here somewhere as well. Inside this, as you will see in the previous lectures, there are many different body cavities that have developed surrounding the future, or preparing the body cavity within which will develop the respiratory system, the circulatory, the heart anyway, the cardiac part of the circulatory system, and the digestive system. Then in week 5, there's a rapid development of the head, the early kidneys, paddle-shaped upper limbs. So everything is just going to proceed to grow and develop and become more recognizable as a human organism as everything becomes more complex and pushes together to coordinate and orchestrate this development. In week 6, the upper and lower limbs, especially the upper, rostral develops first, will become more and more apparent. Elaboration of eyes and ears, so we're seeing a lot of things coming together in the area of the face and the neck to produce recognizable structures. Little digital rays on the hand become present and recognizable in the forelimb before the hind limb. The heart bulge is very prominent. Week 7, we can see obvious digits in the upper limb buds. Small abdominal cavity will accommodate some of the rapidly growing intestine, and the rest will herniate into the body stalk. Now has become the umbilical cord, so there will be a new obvious bulge. And then beginning of the upper limb ossification in week 7, so the laying down of the cartilage that will begin to ossify has occurred before then. So we see the week 7, stage 19, Carnegie stage 19 embryo here with obvious signs of digits developing or ready to develop in the forelimb and the hind limb. And there's a little bulge in the umbilical cord where it is attached here. That's the herniation of the elongated intestine into that area. Until the abdomen gets large enough to bring it back in, that bulge will be present in the umbilical cord. So in 6 weeks, obvious digits in the hind limb and forelimb. The ear is developing, eye is developing, nose is developing, so the development of the facial area is highly complex. Week 9, well-defined digits separated by webbing, so a lot of cellular communication going on here, telling certain cells to die through apoptosis to separate the digits, and then those that remain within the digits to differentiate into the musculoskeletal and cutaneous systems. So there's purposeful limb movement, so we already have the cartilage and some ossification for joints, musculature, tendons, ligaments, and obviously neural innervation. The tail is going to become greatly reduced, and the lower limb ossification will begin in week 8, and we see that here in this slide. The limbs are very obvious. You can see the shoulder, elbow, wrist, the hip, knee, ankle very prominently. It's distinctly human. It's disproportionate. The head is half of the embryo. There's some external genitalia developing, but you cannot determine the sex, so it's indeterminate at this point. And here at Carnegie stage 23, 56 days, this is obviously a human being, or definitely a primate that we can recognize. I appreciate your time and attention, especially being so patient to wait for me, and the extra few minutes to complete this lecture. So on Wednesday, I will come in to initiate the lecture scheduled for that date on the syllabus. No class Wednesday. Health Professions Fair. Show up. I'll have to go and take a look. I can't remember if I have very many questions on that or any at all.
Good news to tell you.